Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week on Careers in Discovery, we spoke with Graham Dempsey, Chief Scientific Officer of Quiver Bioscience, a fascinating company combining stem cell models, optogenetics and machine learning to study brain diseases at an unprecedented scale. Graham's had an interesting career and has risen from senior scientist to CSO in one company. And we talked to him about that, as well as the realities of growing a biotech, the challenges of translating research into real-world impact, and why persistence is key to success. I'm delighted to be back on Careers in Discovery with Graham Dempsey of Quiver Bioscience. Graham, welcome to the show. It's been a long time coming.
Yeah, Tom, great to be on the show, and thanks for the invite.
Of course. A bit of background. Graham and I have been back and forth battling various bugs to record this podcast. So this is third time lucky in the schedule. But yeah, looking forward to diving into your career. We've talked, Graham, in the past about the work you're doing at Quiver, and it's fascinating stuff. Can you give us the sort of the overview, the headline of what you're up to over there for first to start?
Sure. Yeah, happy to do that. So at Quiver Bioscience, we are very much focused on creating a new approach to drug discovery for the brain. Mm-hmm. So if you think about what makes drug discovery challenging, in particular in neuroscience, is how do you create models and systems to be able to study diseases and the impact of therapeutics? Mm-hmm. It's traditionally very challenging for the field. If you look at the success rates in our industry, neuroscience, drug discovery is among the harder things that you can, among the harder endeavors you can pursue.
Yes.
So Quiver is very much focused on creating scalable model systems that allow us to study the brain, and we have a variety of technologies that we bring to bear to address those challenges, which happy to talk about.
Yeah, it'd be great to talk about that because you have a really interesting combination of in silico and wet lab technologies, like the platform that goes from the computer into the real world. So it'd be great to talk about that a bit more.
Yeah, happy to expand on that. So typically when you want to study the brain, it's very difficult, more difficult than other tissue systems because you don't necessarily have direct access to the relevant tissue and the relevant cells. So one thing that we have been focusing on is using technologies that allow us to create models of patient brains using stem cell technology. So that's one important component, which is from a blood sample from a patient, we can differentiate stem cells into different types of neurons. As you know, the brain has a diversity of cell types, and you want to make the right cell type to model the disease of interest.
Yes.
So we've come up with a variety of ways to do that and to do it scalably, meaning that we can make many billions of cells for the purposes of studying a disease. Mm-hmm. So that's really part one. And now, importantly, though, that's one part of the story. If you want to understand diseases of the brain, as you know, the brain is an electrical organ, so it is a careful orchestration of electrical information and chemical information. And as you would suspect, those are the aspects of the brain that are perturbed in diseases.
Right.
So if you have pain, it's associated with changes in excitability in your sensory neurons. Mm-hmm. If you have epilepsy, you have misfiring of excitatory and inhibitory cells. That balance is shifted. So ultimately, it's the electrical information of those cells, which is a critical indicator of the disease. So what we can do with these scalable patient models is we can look at the perturbation of different genes and what it does to the function of neurons.
Mm-hmm.
And that is entirely enabled by our inventions in a field called optogenetics.
Right.
So what does that mean? Ultimately, what optogenetics allows you to do is to convert electrical information into light.
Yes.
And light signals are inherently simpler to detect. They're scalable. You can look at many different colors because of, of the spectrum of reporters that are available. So it's a very versatile technology. You're all familiar with, because you've almost certainly used a microscope at some point in your research. So you know that it is light-based technologies are incredibly powerful of the information that you can glean from biological samples. So we've been our team here at Quiver, in addition to neuroscientists and biologists, we have engineers. So we have hardware and software engineers. We actually design and build custom instrumentation to be able to take these cellular models that we create, essentially disease in a dish models,
Yes.
and then to use these optical tools to then interrogate and say, okay, what is changing in those neurons in a disease? And how can we use that information to inform drug discovery?
Yeah.
And this is the third element of this, which is the computational part that you referenced.
Mm-hmm.
Because inherently what we do is we generate massive data sets of the function of these disease model cells. So now the question is, okay, how do I extract quantitative information from that data to actually inform drug development and drug discovery, right? And this is really where we are very excited by modern compute methods like machine learning. And these are perfect data sets for deploying those tools. They're very highly annotated, high quality data sets that allow us to then say, okay, if I take this data, I can learn what are the important aspects of the firing properties of those neurons that are changed in the disease. And different diseases, as you would imagine, and as we've learned, change neurons in different ways. And that ultimately is the basis for how we then can evaluate a therapeutic candidate. So, and then ultimately feed that information back into how we design those molecules. So it becomes a lab in a loop type process where we can build data sets. We can use advanced compute to learn and design, and then we feed that information back, and we can test our hypotheses and test in the lab what actually happens. And we think that's a really important component of bringing together compute and experiment.
Yes. And there's a couple of things that have struck me about the work that you're doing. And the first of which is something I've never really given that much thought to before, but I'm sure you have. A lot of I guess traditionally a lot of our observations about neurological disorders and neurodegeneration and things like that have been based on post-mortem observations, right? And so you lose a lot of the chemical activity, but you also lose all the electrical activity, right?
100%. That's
so
right.
Actually, being able to do real disease biology on what happens in these conditions is critical, right? To figuring out the mechanism.
I would say that it is essential, frankly, and I think that is an area that we have really invested in is creating ways to interrogate the disease biology in entirely new ways. You mentioned neurodegeneration as an example. You want to be intervening, you want to be understanding the disease not after the neurons are gone, but actually before, and almost certainly, as you might expect, before there are changes in the electrical properties and the synaptic properties of those neurons in the brain. And we want to be able to model that and use that to inform our drug discovery efforts.
Yeah,
absolutely.
So you can treat people before they die, ideally.
Exactly. That would be exactly right.
Yeah. And then I think you referenced this when you were describing the first element of your platform. But one of the things as well that we've talked about before is that the scale at which you're operating this platform is greater than anything else out there, right?
Yes. Exactly. So, typically in neuroscience, the gold standard for studying the electrical properties of cells, is generally relied on manual intervention methods, and this is a place where optical methods are particularly powerful. So rather than having to physically interact with every cell one at a time, we can let the photons do the work, for us. And what that allows you to do is to actually study hundreds of thousands of neurons per day per instrument with single cell and single action potential, single spike level resolution.
Yes.
And that scale is just not accessible with manual methods. So then you can imagine now that you have a path to create many billions of neurons or more, and then you have a way to interrogate them in response to a disease or in response to a candidate drug molecule. You just couldn't do that with manual methods, and that is, I think, an important component of what we do. And I would say maybe the really interesting outcome of this, is that we're creating an entirely new data layer for our industry, for the pharma industry. In many ways, because people just didn't have access to this scale of neurophysiology data.
Right.
So that means that we believe that there is important information embedded in the functional properties of those cells. And we think that, importantly, that's a bridge that hasn't existed, because when you think about typically how drug discovery is done, you have molecular targets, you have hypotheses around what you think the right disease molecular target is. We have really wonderful ways to develop molecules against those molecular targets, an embarrassment of riches of different modalities. But then the question is, what happens in neurons, in intact cells, before you start to intervene in intact organisms and in vivo? And this is really the gap that we're filling with our platform.
Yeah. I've got so many other questions, Graham, but I'm conscious of time. And we'll come back to talk more about the work you're doing there, because you've been with the company through various iterations, and you've been in lots of different roles in the organization. So as we talk about your career, I think we'll touch on a lot of this again, I'm sure.
Yes.
I always like to go back to the beginning, though, and back to the sort of origins of people's careers. Tell me about why science, why this branch of research, why drug development, why this career for you, do you think?
Yeah, I obviously have been reflecting on this quite a bit these days, but I would say that definitely not a linear path for me, and I think that's almost certainly true for most people. That is to say that depending on how far you go back, but certainly I think I've generally been a math science brain since I was young. But in terms of getting to this area of technology and biomedicine, originally when I kind of started on my path, I knew that I liked math and science, and medicine was really interesting to me for a variety of personal reasons. But the real story is that my dream was to be the team doctor for the New York Giants. And it's been a tough decade, but in any case, that was something that I was really excited about. Was getting into medicine, but in the context of some of the sports and athletics that I was really excited about. So I started in college on a path to sports medicine, thinking that was going to be my trajectory. I would go to medical school and be a clinician. And then, I started and actually started my freshman year at a small liberal arts school called Springfield College here in Massachusetts. Wonderful people. Ultimately, though, I realized, I really want to try research, and I want to actually give it a go. I think it was really interesting and exciting, and I think I wanted to find the right opportunities to do that. So I actually transferred out of Springfield College to the University of Pennsylvania, so UPenn in Philadelphia. Both for resource reasons. I think Penn is as people know, is an amazing university with just outstanding research at many different levels, and so I wanted to give it a go, and got there, and as the story goes, I actually wanted to find a way to get into a research lab.
Yeah.
So I cold emailed many dozens of professors because I just got there, and obviously have very little to offer other than sort of washing the dishes, which is what I did in the early days. But by good fortune, I actually heard back from Yale Goldman's lab. He was at the Penn Muscle Institute at the time. An amazing professor, electrical engineer by training, but actually a clinician.
Right.
Started in his lab, and I sort of did anything that they asked me to do. But then ultimately, what was incredible was this lab was really cutting edge when it comes to this interface of advanced technology and measurement technology,
yes,
and biomedicine. And Yale, he's just amazing because he would be lecturing to the on cardiac biology to the medical students, and then he would come back into the lab, and we would have one of our sensitive detectors on the instruments was broken, and he would sit down and fix it because he was an electrical engineer. I just was like, wow, this is really super exciting. So that really got me inspired, and I was doing biochemistry and biophysics at Penn. It's very much this interface of biology and chemistry and physics and computer science. It's just like so interesting and so exciting. So I had an opportunity to do research there. I got a chance to meet Roy Vagelos. I was part of the Vagelos program. Roy Vagelos, former CEO of Merck, one of my professional heroes. It was an amazing experience, and from there, got to do real research and publish a couple papers. I said, okay, well, you know what? I want to do this for real. And that led me to apply to PhD programs. And I ended up at Harvard Medical School in the biophysics program. And, as Harvard, like Penn, amazing, just incredible scientists and researchers, really led me there. And it was there that, I started and ultimately did my PhD work in the lab of another incredible biophysicist. Her name is Xiaowei Zhuang, in the Harvard Chemistry Department.
Yeah.
And this was the early days of a technology called super resolution imaging, which sounds really cool, doesn't it?
Yeah, it does.
Yeah, it does. And it's really, it was taking light microscopy, which has traditionally been thought to be limited in its spatial resolution, meaning the types of things that you can actually resolve within a cellular sample.
Yeah.
And showing that you can actually achieve nanometer scale spatial precision with an optical instrument.
Yes. Okay.
And that is this was thought to be a limit of physics, and it turns out it's not. Several groups were developing similar alternative methods, and the field itself actually won the Nobel Prize in I believe 2014 chemistry.
Yeah.
So that was, that was sort of really exciting, and then that was really the launching point into commercial science, because that's where we ended up commercializing our technology with Nikon Instruments. Just an incredible team that I got to work with within Xiaowei's lab to do this. It's very much a team sport. And I said, wow, this is, taking academic science that is super impactful and exciting and thinking about how you could broadly apply it commercially was a major launching point for me.
And I'm curious. This might not be something you've ever thought about, so I apologize if this becomes a difficult question. But
No
I
problem.
think, in biology, it's kind of accepted, I suppose, that there's a lot we don't understand, and the things we do understand might not be correct. And there's not so many laws of biology in the same way that there are laws of physics, right?
Yeah.
And you worked in a group where you, as you say, you were expanding the limits of what was thought to be possible
Yeah.
in physics from a physics perspective. Do you think that has had an impact on how you view research and sort of how you view the work that you do?
100%.
Mm.
Probably every day
Right.
that I do the work that I do and that we do together. And I think that was, for me, that was something that was so exciting about
Yeah.
the field because, biology, obviously biology and our understanding of biology has grown in leaps and bounds for sure.
Yes.
But it's really the marrying with physics and technology and computer science that I think is going to help us understand the rules of biology with much greater precision. It's not as clear of a deterministic science at this point, but as we get better, in our ability to measure and understand what's happening at a very fundamental level, we focus on the cell being this fundamental unit. I think we are learning, and we are going to continue to learn with much greater precision about the biology, about biology in general, and certainly human biology of the brain. And that ultimately we believe is going to inform how we then create medicines.
Yeah, absolutely. And I suppose I suppose we live in a world now where we're constantly re-examining the things that we perhaps thought were possible and not possible, right? And so,
for sure,
bringing that mindset to the work that you're doing is, I guess, empowering in lots of ways. It's probably has its days where it's where it's challenging as well, I'm sure.
Yeah, very, very challenging, and that's what I think makes the work so compelling. And we enjoy the challenge for sure. And I think that is acknowledged in drug discovery. We've continued to make amazing progress in creating ways in which we can drug different targets. There's just so much beautiful work being done there. But I think it would be broadly acknowledged that we just don't know enough. We need a deeper understanding of the biology of disease, because ultimately that will then help us make better decisions in terms of how we deploy those different molecules to address diseases. And I think I'm really excited about that. Absolutely.
So going back to your career, Graham, so you had this experience working with commercializing a technology, and that gave you a bit of a taste for working in commercial research. And was that because of the sort of, you saw the practical implications of it, or
Yeah, I think it was broadly acknowledged, the practical implications. If you think about it, almost every laboratory uses a microscope in some capacity, and light microscopy tools. The advent of fluorescent proteins is more or less ubiquitous in research around the world. So certainly having instruments that give you a much greater level of spatial precision would be broadly of interest, and allow you to then again learn things that you couldn't do before. So it definitely acknowledged, and there were a variety of instrument groups that did commercialized versions of these technologies that are now deployed in labs around the world today. So I think definitely recognize the potential impact for sure.
Fantastic. And so you tell us about then the steps that you took into the commercial world and into the biotech industry, and talk us through that journey.
Boy, what an interesting, it has been quite quite an interesting journey. So, it turned out that down the hall from where we were doing this work and across the courtyard were two, also incredibly talented professors. One, Adam Cohen in the physics and chemistry department, and then Kevin Egan in the Harvard Stem Cell Institute, who in their own rights were developing both stem cell models of brain diseases. So how do we use stem cell technologies? Because you can now start with just a blood sample from a patient to create a model of their neurons.
Yes.
And then Adam's lab was developing technologies around optogenetics, which are, as mentioned, ways to use light to interrogate electrical systems like neurons or cardiac cells.
Yes.
And, to do that, really required bringing, sophisticated biology and physics and computation to the table to do it. And it was very, very much realized early on, well, okay, you have the ability to create scalable cellular systems of the brain, and we have ways to now interrogate them at a profound scale. These two things make tremendous amount of sense to put together, and a desire to say, okay, well, how can that then be used and deployed in the biopharma industry, and frankly, even beyond that. So the company was founded. This is around 11 years ago, called Q-State Biosciences, and I actually was, as I mentioned, down the hall from Adam. Adam was on my PhD thesis committee, so I got to know him and the incredible people in his lab, and then Kevin as well, and then David Margulies, who's a serial entrepreneur in the health tech space for many, many years. It was decided to start a commercial enterprise to bring this technology to the marketplace. And I joined as a senior scientist as a first initial full-time hire of the company. It was a lot of fun. It was you do everything when you start, and there's only a couple people, and it was really building from the ground up the translation of the academic science, which is it's powerful, but it's raw, into a industrialized commercial grade system that you can use in applications for drug discovery for neuroscience. So that's how it started, and yes. Then happy to talk about the ensuing years.
I think there's some interesting things that I'd love to explore, because so Q-State became Quiver eventually, right? And so it's the yes. same organization, but I know like any company with any longevity, it's not always been up and to the right? There's been a few turns along the way, so it'd be great to hear about that. But also, sure. I think it's not common that someone joins a company as a senior scientist and becomes the chief scientific officer and has that consistent progression through a company. So really interested, I suppose, in, as the company evolved, the things that you learned along the way that have enabled you to take on more responsibility, take on a bigger role, and be effective as you've grown.
Yeah, I mean, it's just I have so much gratitude and feel it's such a privilege, the situation that I have been in. And, I would say that, as mentioned, started senior scientist, building instruments and building the platform. And then over time, as we, started to get traction with working with commercial partners, starting to take on more responsibility, beyond sort of hands in the lab and into managing teams and organizing teams and efforts to to deliver and execute on the work that's super important.
Yes.
So a tremendous amount of learning along the way. I would say that when you start with technology system that has many potential applications. I'm sort of focusing on drug discovery, but
yes,
even for basic research, these tools can work in other excitable cell types. So we chose neuroscience for a reason. You have a potentially very wide aperture, and you've got to figure out who you want to be and where you want to focus.
Yeah.
And to be frank, and as I think many would tell you, it's never a straight line. So you learn a lot about. Importantly, what we wanted to do is learn about where can the tools be deployed effectively? Like where are the places where we think we have real traction first, and to focus on those. And I would say that the early years of the company, we were really trying to understand how we can both enable ourselves in terms of our ambitions around drug discovery, but also how we can use these tools in collaboration with pharmaceutical companies, because we want the impact of the technology and the tools to be as impactful as it possibly can, and that means that you want to deploy it not only for your own purposes but for collaborative purposes.
Yeah. So
I would say that there was a sort of a growth trajectory there to learn and get there, and then I think learning how to organize and work in teams. This is like one of the things I just really love about working in commercial science is it's a team sport,
right.
And especially when you have what we have, which is a variety of technology disciplines, biology, physics, etc. You really need a team. You need a really high functioning team to be effective. And I love that. I love that. First of all, building those teams and then leading those teams is, again, one of the great privileges I feel.
Yes.
But you don't necessarily learn those things in graduate school, right? You're learning how to be an independent scientist. So I think you're learning about management, and you're learning about organizing teams, and then also just thinking with a commercial mindset, because often when you're in an academic science setting, you're often very interested in very basic science. That's super, super important, by the way, which we need. We need that. But then in commercial science, where you're trying to take the learning and actually develop products, either therapeutic or other. It is a different mentality, and it's something that for me, I learned over time, and I continue to learn. I continue to be a student of this industry.
No, of course, and so lots of sort of categories of learning, I suppose. Learning about working with other people, working with teams, running teams, but then learning about the working with external partners, the commercial side of things. I imagine, investor relations, all that kind of stuff has come along with it. And then you sort of part of a executive team. That's a different thing again.
Exactly.
And just growing an organization, right? Growing a company, there's always new things. If you bumped into yourself 11 years ago, Graham, as you were joining what was Q State at the time, what do you think? What are the things? Maybe one or two things that now that you would pass on to your younger self.
That's a great question. And I would say that maybe one thing is maybe to help smooth the line a little bit in terms of the trajectory is to try to get to focus as soon as you can in terms of what you are effective at, what your technology is effective at, and try to capitalize and do that as efficiently as you can. It's a balance, I would say, because things do take time to really learn and progress. I think everybody generally wants the benefit as quickly as possible, but look at the success stories that you see, and these things really do take time and learning. So often, if I run it myself, I certainly I would say that, given where we were at that time, I think there are areas of focus that we've learned where we're really good now, and especially how we can bring advanced compute to our tools. That I would say, I would really shine a light on that.
Yeah.
For my younger self to say, okay, this is where you should spend your energy. And then maybe another thing is to say, being a student of your industry is super important.
Okay.
Something that I encourage everyone. So everyone at the company, encourage them to, be a student, to learn about the industry and be knowledgeable about our industry, because first of all, it's super interesting. Intellectually very interesting, but importantly, that's kind of how you can keep your finger on the pulse of what's going on. And I think everybody at a company should do that, because that's the industry you operate on. And I would say that, for again, talking to my younger self, I would say, would shine a light on that as well, and try to be more aggressive in that regard in terms of really learning about the industry, and the focus area that we ultimately would settle on.
Yeah, absolutely. I think the first point you make there about things taking time and
yes.
being focused over time, and I guess the sort of compounding benefits of that, right? I actually I was writing a blog about something the other day, and I asked ChatGPT to work this out for me. So obviously we know that, like, you get 1% better at something every day.
Yeah.
And the standard thing is at the end of the year, you're 38 times better than
exactly.
you were. So I asked it to work out a decade. And it's 1.17 quadrillion times better.
Wow. Okay. Well, there you go. Well, I'm going to use that. I'm going to tell my kids that.
Exactly.
How much better I am now. Yeah.
Well,
I couldn't agree more. And you sort of mentioned, like, okay, this is a place that I've been for more than a decade. I'm personally very passionate about what we're doing, what I believe we're doing. But the compounding that comes with that time, and you'll find that the team here are a lot of the same faces that have been here for many years and have been through the many years of the company. There is just so much that comes with that compounding, both from as it relates to the intellectual requirements of the technology and the science that we do, but also just working effectively with people on your team, because you become, you really understand each other. You understand each other's strengths and weaknesses, of course, and you can play to that just like any effective team does. So that I think has really been important to me, and understand that it is an approach to a career, but frankly, if in this environment and what we do, to get to success and to make an impact, if it takes many years, that's okay.
Yeah, and I think it's really important to hear that amongst. I think we have unrealistic expectations of businesses and what success means in business these days, right? If something's not a $100 billion company in 3 years, then what's the point, right?
Right.
But and there are companies that do it, right? But but most don't, and there's lots of really successful businesses that are doing important things, that are doing them well, that are providing value, but also that are providing careers for people, that are providing development for people, and advancing the field and doing all those sorts of things that, maybe they're not growing at 300% a year, but they're growing and they're making progress and they're moving forward. And I think it's important that, now we're 2 years out from 2021, 2022, right, where all that was going on. But we maybe are at a point where we should reset some of those expectations and realize, actually, these are really good businesses that are making real dents in the world.
Yeah, I completely agree with that. And I think you have to decide on what's important to you and what your North Star is, and we want to bring the most impact that we can. I mean that's why we do what we do is we get a chance to interact with patients and see the what they go through, and we have an opportunity to do something real and impactful, and so we want that to be as broad as we can. But as you said, it's our growth trajectory, and even as we've been continuing to make progress, we really believe that we are advancing the field and advancing knowledge and understanding, and there are even greater opportunities ahead of us right now.
Yeah, absolutely. So as much as you can, I appreciate some of it you won't be able to talk about, but tell us a bit about what's next.
Yeah, of course. Happy to do that. So, we are, as a company, you mentioned kind of growth and our trajectory. And so very much fundraising, as I suspect most, everyone's in fundraising mode at some level. We are as well. We're raising a Series A financing round, and we want to be able to use that capital to now advance molecules as well as our platform. So we started with, again, an empty room and an empty table. Over the last years, have been building the platform itself and all the infrastructure to support that. We've learned a lot about where we believe we can be immediately impactful in areas like chronic pain and neurodevelopmental disorders, to name two, and neurodegeneration. So now we want to use the tools to bring molecules to ultimately to patients. So that's really the next part of the next growth for us is to progress therapeutics forward, and we're excited by that. And then for the platform, as somebody who's been in the technology space, you have to evolve and you have to keep up with the evolution of the science, and we want to be at the forefront of that, and we will be at the forefront of that. But that means investing in all the things that I talked about in terms of how we model diseases, about how we understand what happens in disease, and expanding the scope and opening the aperture of that, and then bringing in computational methods to not only advance our own unique data layer that we create, but to start to and continue to connect what we do to really these vast multimodal data sets that are being generated around the world at places like the Allen Brain Institute and CCI and others. So being able to bring our data layer and the insights that we bring, and certainly connect those and the learnings to other data types and other approaches, we think is going to yet further deepen our understanding of the diseases. And then, for us, we want to bring. We see the platform as having the opportunity to not treat disease, but to potentially treat many diseases. And there is tremendous need. I can tell you, as I'm sure you know and others know, there are a variety of diseases that need innovation, and they need therapeutics because we have sort of antiquated approaches.
Right.
So we want to be able to use the scalability to accelerate the rate at which we can bring therapeutics to the marketplace.
Yes.
And that is going to be important as we start to take what we do and really in the sense of creating a factory of sorts to be able to generate medicines for the marketplace in a variety of different diseases. And to do that, we have to invest and continue to develop, and that's what we'll be doing.
No, absolutely. Well, I think you're taking a really unique approach to a very important problem, and I'm fascinated to see what happens next. And it's an important time, exciting time for you, and we wish you the best of luck.
Thank you, Tom. I will say that there's a saying, the difference between obstinance and persistence is success. So I hope that the next time we get a chance to talk, you will say how persistent we were, in fact, through all the challenges, and we look forward to continuing the work that we believe in.
So do I, and we'll be keeping our fingers crossed for you.
Thanks, Tom. Appreciate it.
Thanks, Graham. Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.